All right. So it should be recording. Hey, Mike, can you give me a quick confirmation you can hear me?
What's that?
Oh, that's it. Just need to hear your voice. All right.
All right. Hello, everybody. Welcome to Bryan Duoto's thesis defense. Welcome his family, friends, colleagues, and of course the thesis committee. Thanks everybody for coming. So this is my opportunity to introduce the candidate. Bryan has a BS degree in biology from Sonoma State University. He then completed a master degree in molecular and cell biology and microbiology it was a joint program between Stanford and San Jose State University. He then got industry experience working for Juno Therapeutics in Seattle, first as a scientist and as an engineer. And then he thought he comes back to the university to join us and pick up this nice green jacket and black t-shirt to work in our lab. He's finishing or has finished, very close to submission, was submitted 2 publications and also a patent. And if you know Bryan well, you know he's curious about science and scientific discoveries, but what he really cares about is translation and developing things that could make a real world impact. So I think he's particularly proud of the patent. And what he didn't know quite yet is that there's actually a company interested to license it. So we can talk about that offline a little bit more later. Bryan had the opportunity to attend conferences, and I think when you look at these pictures, he picked very wisely in the locations. So first meeting was in the nice mountains of Snowbird in Utah for the Foundations of Nanoscience meeting. And then I think the family also came to visit the quantitative biology meeting in Hawaii, maybe not so much the meeting, but the location. So being at the FNano meeting, Bryan is always eager to talk about science, not just in the conference room with Professor Zhao, but also on the lift, having lots of discussions every time we're going up the lift with Professor Garman, myself, and others about computational analysis of RNA sequences. Bryan's hobby is computational biology. He didn't get to do that very much in my lab, so he plays around with the lab logo, made these nice animations. He also is leaving behind sort of a legacy to the lab. Thank you for that, the little Steinmetz, our virtual postdoc. So he trained a language model, an AI model on all the publications that we've published. And so now we have a virtual postdoc that we can ask questions. So this project is still sort of in the final stages, but yeah, thank you for the initiative and the effort of putting this together. Here just a couple of fun pictures from the lab. Right, the first picture, I think, post-pandemic, when we could be together again, and then a happy moment when we moved into Franklin Antonio Hall. I think Bryan has grown quite a lot as a researcher. He came to the lab being technically very skilled, showing that he's already worked as a scientist and engineer. But I think he has really grown in terms of working independently, designing projects. And I think with his career path, maybe starting his own company or being in the biotech space, that he will do really well. I think his biggest fan is probably his daughter Madrone, who's also in the back there. Hi, Madrone. And Bryan is always a very friendly person, a very happy person. But when he sent me this picture, it's like such a nice smile that I thought I end on the slide to introduce him, to make him feel good and comfortable for his defense. Thank you, Bryan.
Yeah, thank you. No, I appreciate that. I just wish I had shaved a little bit more for this picture because you can't really see my mouth too much, but I am happy under there. Definitely happy. So let me just switch over to this. So I'll get right into it. Thanks everybody for coming today to my thesis defense, whether you are virtual or not. Actually, let me just make sure that nobody's in the waiting room real quick. There we go. So I have my in-laws, my dad, a few other people on the call today, so they'll be listening in. They can't make it today. My parents actually run a school and they're doing graduation today, so a lot of graduations happening right now. And my younger brother's graduating from Santa Clara University in the Bay this coming week as well. So combined graduation party. Okay. So today, what I'm going to be talking to you about, it's a little bit different than actually the posting for the department. So I talk about it being an immunotherapy platform, but in reality, we're just going to really take a step back and look at potato virus X or PVX, the main subject of my research, as a gene delivery platform. So retooling a plant virus to essentially work as a gene delivery technology for mammalian systems. Hold on. Come on. They're having some issues earlier, so let me see if I can fix this. There we go. So my committee today, there are most of them are here today. Unfortunately, Liangfang Zhang can't make it today because the vice chancellor is calling all the department heads for an important meeting. But we'll be meeting on Friday at 9 a.m. So while I might get a 3/4 majority today, don't call me doctor until Friday, hopefully. But I first want to introduce Dr. Nicole F. Steinmetz. She's my chair, or Steinmetz, I should say, sorry. She's my chair, my advisor. She's been the champion of a lot of weird ideas that I've come up with, and I just really appreciate what she's done for me and that great intro she gave me earlier. And then in the committee today and on Friday, we have Doctors Nisarg Shah, Liangfang Zhang, and Prashant Mali, and they're sitting in the front row with me today. All right. So kind of getting into why I'm doing this project, what's the importance of this project? So a big issue that we have is getting different nucleic acids, I think RNA and DNA, into cells, right? That can be used for a lot of things. One that you might be familiar with is COVID-19 vaccines. These took mRNA and lipid nanoparticles as a vaccine platform, injected into muscle tissue, and delivered mRNA into cells, right? So the alternative to that is delivering DNA into the nucleus of cells. We won't be getting too much into that. But how can we further improve upon kind of that platform, getting mRNAs into the body of the cell, the cytosol of the cell, and overcoming those barriers of getting inside the cell? So currently, these are kind of the different methods that you see, and they come in a lot of different flavors. So we have physical methods for getting inside the cell using lasers called optoproliferation. We have polymers that are more of a chemical method, electroporation, which is applying a bulk charge to the cell and opening it up for delivering payloads. And then we have microinjection, which is using microneedles to poke holes in the cells and deliver payloads directly into either the nucleus or the cytosol. Now, the two biggest technologies are non-physical means that we use a lot in the clinic today. One is lipid mediated. So think COVID vaccine using lipid or fatty particles to encapsulate our cargo, delivered and fused to the cell membrane and get mRNAs into the cytosol. And then the one that we see in our most advanced therapies, which is viral transduction. And this is using live viruses, adenovirus, adeno-associated virus, lentivirus, to get advanced gene therapeutics into either or the cytosol or the nucleus of the cell using the natural ways that viruses infect us and translocate their cargoes to these different spaces. Yeah, it might be somebody at the door right now. Okay. I'm gonna have to click through this. And so why do we choose this candidate for this space that's already kind of crowded with a lot of solutions, right? Why a plant virus, and what advantages or potential do we see in that as a nanocarrier or a nanotechnology for delivering these payloads? So if you look at the structure of potato virus X, you'll notice that it's a filamentous virus, which is kind of a rare morphology or shape. And it's about 500 nanometers long by 15 nanometers in diameter. You can see a couple nice pictures of it through TEM on the upper left hand. And on the upper right hand is a cryo-EM of what the structure looks like. So it has 1,300 identical coproteins. So the outside shell of that virus is the same repeating unit of coprotein. And it encapsulates a 6.4 kilonucleotide genome. So 6,400 nucleotides. And it is pitched in a 3.6 nanometer helical array. So it's helical, kind of like what you'd see with DNA, kind of that rotating, spinning pattern. And there's about 8.9 coproteins per turn of that helix, and 45 nucleotides on average per turn of that virion as well. So where have we seen some promise with PVX? So we've done a couple studies in our lab. I won't get too deep into them, but essentially in the upper left hand, you can see that we can load the outside these coproteins with different drugs like doxorubicin. We can use it as a therapeutic in treating different types of cancers. Here we have lymphoma, and you can see there's good survival rates when we use conjugated PVX with doxorubicin versus free doxorubicin or the controls. And on the right side, you can see when we do an in vitro assay that PVX, when labeled with Cy5, which is a dye for tracking the virus, it binds to a lot of cancer's B cell lines. So we can see it binds to Raji cells and Dowdy, while leaving a lot of other cell lines that are less specific or healthy cell lines alone, including normal B cells. But we do see a good amount of cell binding events happening between the virus natively and these different cells in a dish. And there's a couple other things that it has going for it. So when we talk about it from a materials perspective, it's got a high aspect ratio, which means it's got the potential for a lot of loading. It drains to different tumor environments. It has good tissue penetration. And it's morphologically uniform. So when we talk about polydispersity versus monodispersity, it's a very monodispersed particle. It's usually about 500 nanometers plus or minus 10 nanometers in terms of length. And it's water soluble. It's a highly homogeneous soft matter. And you can create it through a lot of different manufacturing methods like molecular farming, which is basically infecting a plant and purifying the virus, or fermentation, making the coproteins in the RNA recombinantly through E. coli or bacteria and cell-free systems. All right. So how can we determine whether or not this is a good place to start for being able to carry nucleic acids, right? So I mentioned before it has a single-stranded genome, that's 6,400 nucleotides. What I didn't mention before is that it also has a 7-methylguanosine cap and a polyadenosine tail. Now, those two features are really important because they're the exact same features that you see on mammalian mRNA. So when we talk about human mRNA, it also has a poly-A tail and an M7G cap. And our mRNAs come in various lengths, but 6,400 is already a pretty long starting point. And it's evolved to be in a lot of ambient conditions too. So these viruses, they're out in the world, they're infecting plants. They have to live in different humidities, acidities, you name it. And that really eliminates a lot of cold chain needs that we see in things like lipid nanoparticles that need minus 80-degree storage, right? So it overcomes a lot of the hurdles we see with similar technologies, and it demonstrates some good features in terms of being able to encapsulate nucleic acids. The one I will call the most attention to in this slide is in the circle here. It's called the stem loop 1. This is at the very beginning of the genome, the nucleotides about 48 to 84. And in this stem loop is the binding site for coproteins. So this is the nucleation, the origin of assembly site for PVX. And we saw in some papers from the early 2000s that you could utilize this RNA structure to drive assembly in non-native RNAs, so synthetic RNAs or partial RNAs of the genome. All right, so just a few hypotheses based on what we've seen so far, and a few generated that I'll add some more evidence to later, is that we have this system that can recognize an RNA sequence, so we can hopefully engineer that OAS sequence onto any RNA and get assembly in vitro. And that these virus-like particles derived from this method are able to encapsulate different single-stranded RNAs from different lengths and different classes and form particles of possibly different sizes based on the RNA length, because we saw that there's about 45 nucleotides per turn. And that these coproteins that we're using to make these virus-like particles can be engineered recombinantly to express other proteins, to bind to different linker molecules and undergo bioconjugation. And that cargo within these VLPs can be delivered to the cytosol cells safely while being protected from nucleases out in the environment and in a dish. and that the delivered payload actually turns into the desired protein, right? mRNA is always, if you remember from high school and undergraduate biology, is always the driver of what we call translation, which is the writing of that mRNA transcript into a functional protein that we want. And then the last thing is that we'll demonstrate that maybe is a little less clear from this introduction is that we can move beyond mRNA and we can go into other classes of RNA molecules that expands the platform as far as capabilities and as far as efficacies when we talk about things like redosing and delivery of different payloads to treat different diseases. All right, so the first thing I'll be talking about today is kind of one of those first hypotheses, which is can we engineer the size and the morphology of in vitro assembled virus-like particles derived from the system? So here's kind of an overview of the manufacturing process of how we make these virus-like particles. So now we're moving away from native PVX and we're making virus-like particles that look like PVX in a dish without infecting a plant. And this is kind of what that looks like. So from the left to the right, we take native PVX isolated from a plant. We disassemble it using a buffer that has a high ion concentration and a low pH. We centrifuge away the genomic RNA because we want to purify the coproteins away from that RNA. And then we feed back in different genomic mRNAs of different sizes so we can see how many sizes we can encapsulate to those purified coproteins. We do an ultracentrifuge after dialyzing into a low ion, high pH buffer to drive assembly. And then we use a sucrose cushion ultracentrifugation at the very end to isolate assembled virus-like particles away from free coproteins. And the idea is that with this first aim, we'll get different sizes of VLPs based on different sizes of mRNA. Some more details about this process. So the ratio of RNA to coprotein to get good assembly outcomes, good yield, good nucleation of assembled virus-like particles is about a 1:50 mass ratio. This means that for every 1 microgram of RNA, we're putting in about 50 micrograms of PVX coprotein. And just some stats on how well this works. When we put in about 1 to 5 milligrams of wild-type PVX as the raw material per purification and assembly reaction, we get about 50 to 85% of that as purified PVX coprotein. It's a 3 to 4 day process for all these different steps up on the screen. And at the very end, we usually get about anywhere from 70 to 90% of that purified coprotein as final virus-like particles after purification. That has a lot of variability, but that's just kind of an average. All right. I gotta stop doing that. We've seen how we make the particles. How do we make the RNA that is going into the particles? So what we did is we took the entire PVX genome and we cloned it into a plasmid up in the upper left-hand corner. We nested it under a T7 promoter so we can make it in a cell-free system using a commercial kit from New England Biolabs. And then we have a BGH terminator on the other side of it so we have a good site for poly-A tailing. We used primers and PCR to amplify different sizes of that genomic RNA so that every single size contains that OAS site, that 50 bases that we talked about in the beginning, and goes anywhere from 250 nucleotides, the first 250 of the genome, all the way to the full length 6,400, and then a couple intermediates in between. Here's kind of what the mRNA looks like after all the steps are done in vitro transcription. So you can see again, T7, a 5' UTR, the stem loop structure, which is native in the gRNA, a gRNA fragment, and 3' UTR in the poly-A site. And after doing assembly, before doing assembly, here's kind of the gels that were run on these different DNA transcripts and RNA. So upper two are DNA, and we can see good bands for every single one of our size conditions, and the bottom, which is the corresponding size conditions of RNA. All right. And once we've made our VLPs, what do these look like? What is the size distributions? What are the outcomes for loading different RNAs into these VLPs? So kind of interestingly, we get two morphologies. When we have under 1,000 kilonucleotides, we actually start to see more of a rod-like morphology, which makes sense. A shorter VLP doesn't have as many coproteins and not as big of a helix to bend and flex like the native virus. But as we get bigger, we get more of that native morphology of that filament shape, right? That bendiness. And you can see a couple of examples of that here. And on the right side, I'll kind of drill down into more of the size per condition in the next slide. But here's just kind of an overview of what each of the size conditions look like in terms of kernel density estimation and histograms overlaid. So we can see that they start to trend to the right as we go from 250 nucleotides upwards. But we only get a sizable shift when we hit the very last condition, which is the native size of the genome, which is interesting because it's demonstrating a preference for its own length of RNA. In this case, it's native gRNA, but we can assume that based on a lot of previous literature and what we're seeing here, that this is a common occurrence for different viruses. They have evolved to look for that size of RNA, despite whether or not it's their genomic RNA. And then if we drill down, here's the different distributions in terms of the nanometers of the sizes of the VLP, so how long they were. And an interesting thing that you see is that contrary to what I told you for native PVX, when infecting a plant and creating naturally occurring virions, is very tight distribution as far as a histogram shape. It's very skinny. It's only got plus or minus 10 nanometers in terms of size. But when we do it in vitro, we actually get more polydispersity and higher order assemblies and multiplets and doublets forming in our different conditions. So that's both a good and a bad thing. So when we look at our medians, they seem to hover around 300 to 350 nanometers with some exceptions. The reason for that being is that when we do the assembly process that I showed you earlier, we're not getting rid of all the genomic RNA from the original PVX when we make our VLPs. So we're actually getting contaminating genomic RNA that's probably adding some noise to our data. The other thing that's happening is that we're most likely getting the incorporation of more than one transcript of these mRNAs per VLP, which is great because it means we have a greater loading capacity for VLPs. And the smaller the RNA, the more RNAs that can likely fit in that helical structure of the virus-like particle. You'll notice that there is a tighter distribution at the very last condition, and we do get that size shift. And the median is actually 616 nanometers, which is about 100 nanometers bigger than native PVX, which is interesting. So there's definitely a packaging efficiency change when we go from in vitro assembly versus native assembly. And we have more polydispersity when we do assembly in vitro, despite the fact that the ingredients for the 6.4 condition are the exact same ingredients that you would see in PVX, that being the native gRNA and coprotein. So just to kind of wrap up this project, we've shown that PVX coproteins can form VLPs in vitro with foreign mRNAs being, in this case, different fragments of the gRNA, not full length, and that we most likely are seeing multiple RNAs per VLP, and that the gRNA from the PVX coprotein purification process is likely influencing the distribution of size that we're seeing in these VLPs. So the next thing I'll talk about is the other engineering challenge with VLPs, which is engineering the outside of the virus-like particle or the coproteins. So we've shown that we can manipulate the payload, the nucleic acid, but let's see if we can get some good outcomes in terms of engineering the outside of these VLPs. So there was a paper and a little bit of existing theory saying that if you were to put a His tag, which is a very common fused protein for recombinant protein production, it allows you to purify your protein with high fidelity and high purity, that if you leave them all on the coprotein, you don't get assembly, you don't get virus-like particles assembling in vitro. And so we thought, what if we just did fewer His tag proteins and we just mixed them at different ratios with non-tagged coprotein? So here we're taking an N-terminus fused 9X His tag coprotein made in E. coli, and we're mixing it with wild-type coproteins derived from the native virus. And so why are we doing this? What is the pros and cons of this? So the first one is manufacturing. We're talking about process plant design, process engineering, tech transfer of different technologies for creating a therapeutic. Having more than one option is good, and having one that has obviously more advantages over the other is good, but having two that you can use simultaneously is even better. So the one that we use in this lab a lot, we call molecular farming. What this means is that we have a walk-in plant chamber We do agro-infiltration of our viruses into the leaf material of our plants, in this case, Nicotiana benthiama. And we take the infected leaves, we blend them up, we purify them through centrifugation and cheesecloth and all these different things. Then we spin them down and then we isolate them from centrifugated pellets. And that's how we typically get our native virus. And that's what you've been seeing this entire time in terms of native PVX. So molecular farming is great. It has a lot of advantages. It's relatively cheap. You need limited facilities, so you don't have to build out a lot of facilities to do molecular farming. The feedstocks and the energy requirements are pretty standard, pretty easy. Water, sunlight, nutrients every once in a while. You can automate it. There's a lot of work that's been done to automate a lot of molecular farming techniques, and you can scale up to large-scale production. You can think of like a growth chamber to a greenhouse to a field of plants. We already have shown different scales of molecular farming because it scales off the technologies that we have incumbent from agriculture and ag tech. So how does that compare to fermentation? So fermentation is already a very, very mature technology when we talk about how we produce proteins recombinantly. So it's a cheaper alternative to molecular farming overall, especially as we scale up. The economies of scale and the techno-economic analyses really point to fermentation being one of the cheaper methods of production for biologics. And it can be scaled up more easily due to existing infrastructure. So every year, thousands upon thousands of liters of bioreactors are brought online across this country. And there's initiatives by DARPA and ARPA-H to expand our capabilities because we see fermentation as a cornerstone of national security at this point. Food security, therapeutics, outbreak response, these are all things that fermentation is very well suited for. It has faster production cycles than molecular farming. You don't have to grow a plant. You just grow bacteria. They can grow at high densities, and we can get a very dense culture of output protein. It has higher product yields than molecular farming, and it has easier purification. So there's no tissue processing steps. We're just going from cells to protein. And very importantly, it has established regulatory pathways. So molecular farming has had some issues with getting through different regulatory pathways to become a standardized manufacturing platform for therapeutics, whereas fermentation has been around for a while. And you can see here just a representation of how we typically do that. We use shaker flasks, we grow up E. coli, we spin them down, we sonicate them, and we isolate away our protein using gravity columns and His tags in this case. And then you can see down here what it looks like to go from the small scale to the medium scale to the pilot scale to the at-scale production. All right. So again, we started this project with cloning in our coprotein into a plasmid. So you can see here, again, we have a T7 for producing this in a T7 polymerase producing E. coli strain. We have our 9X His tag, a way to cut off the His tag called a TEV site, and then our PVX coprotein following that and a T7 terminator. So this means that our E. coli will recognize T7, produce the polymerases, create our protein, and then not produce any nucleases, hopefully to degrade our protein because they're specially made E. coli. And the assembly process, just to kind of give you a modified version of what we saw earlier, the only thing different happening here is we're still doing the first couple steps, but when we purify our coprotein, we're going to be mixing it with different recombinant coproteins from our E. coli strain here at the top, which is a BL21 E. coli strain. And so by doing these mixed ratios of His tagged recombinant coproteins to wild type, we're hoping that we can still get assembly and make these mixed assembly VLPs. All right. And so what did that look like? We try to first quantify this using a gold nanoparticle conjugation technique called nanogold. Sometimes it's also called immunogold. And so what this does is that this 10 nanometer gold nanoparticle kit will that's conjugated to a nickel NTA will bind to the His tag, and basically you can visualize that using transmission electron microscopy. And you can see that down here at the very bottom. So you can see that as we go from the high ratios containing the most His tagged protein, 7:3 being the biggest one, all the way down to wild type PVX, that we have an increasing number of gold nanoparticles per VLP as we go to higher amounts of recombinant coprotein in the ratio mix. And the top right-hand corner, you can just see how that looks quantitatively. So the frequency of gold nanoparticles per condition, as you go up the conditions, there are more gold nanoparticles, up to 8 in some cases for the highest condition per VLP. And one last thing I'll say about this is that the reason why we didn't go above 7 to 3 is because we didn't get meaningful VLP assembly at that mixed ratio. So there is a tipping point in terms of the yield drops, and we just do not get successful incorporation of a lot of his tagged coprotein. And then we also did a Western to also visualize this. So this is called a nanogold, sorry, a Goldie blot Western. And essentially it's the same technology. We're using a 5 nanometer nickel NTA conjugated nanogold particle. And then we're enhancing it by using gold ions from a solution that's depositing selectively onto these 5 nanometer gold nanoparticles and just making bigger and bigger gold assemblies after a couple rounds of application and mixing steps. And you can see here again, kind of more qualitatively, that we get a disappearing of that band at the lowest ratios of recombinant coprotein to wild type coprotein. So some takeaways from this project is that we can, yes, indeed, assemble VLPs in this mixed assembly method. So while we can't do every coprotein in an entire VLP with a fused small protein complex, we can do at least small protein fused to the surface of recombinant ones, and we can accomplish it when we mix in non-tagged coproteins. So again, those wild-type coproteins in this study could be replaced with recombinant coproteins that just don't have that his tag. And we can move on to more diverse classes of fused proteins in the future. So think of things like cell-penetrating peptides to increase delivery efficiency. We can have different small molecules, different proteins kind of conjugate to that surface. And we can utilize mutants of these coproteins to bind different small molecules, large molecules, and linkers to accomplish different aims. All right. And so let's see it work. This is the part of the third objective is the part of these studies that shows that this does indeed work. We can get proteins expressed in the cytosol cells, but how exactly are we accomplishing that? So here's kind of a cartoon of what that process looks like. Starting in the upper left-hand corner, you can see that we do our in vitro disassembly of coprotein. We do our IVT to make mRNA. We recombine them like we've shown previously. And then we can transfect these GFP-containing VLPs into cells, mammalian cells, and get expression of GFP. So why do we use GFP? GFP is a fluorescent protein, so we can see it visually, and we can quantify it by measuring it through different lasers and fluorescent means and photoreceptors. And it's a great proof of concept to show that you are making the protein that you are encoding for in your mRNA. So here's the design of that mRNA. Again, it's pretty straightforward. We have a lot of the same features, a T7 promoter, a 5' UTR, our stem loop 1 structure so we can make VLPs, and then our eGFP downstream of the stem loop 1, followed by a 3' UTR and the poly A tail. So it basically looks like a regular eGFP mRNA, with the exception that we have the UTRs from PVX and we have the stem loop 1 from PVX. Here's just a bioanalyzer or tape station of that mRNA. So it's about 870 nucleotides. So about 1/8 the total size of the genomic RNA. And then here you can just see a quick mRNA transfection showing that the mRNA before loading into VLP works and that we are getting GFP expression in BHK21 cells. All right. So what do the VLPs look like when we do that manufacturing process and that in vitro assembly we talked about earlier? So on the left-hand side, you can see size exclusion chromatography. From top to bottom, we have just the coprotein by itself, purified PVX coprotein. We have the stock virus. And then you can see that the eGFP SL1 VLPs are in the same size regime as the stock virus. So they are smaller. We have a right shift from 8.27 elution volume to 8.63. But it has some small overlap and is expected to be in a similar area as our virions, our PVX virions. Another indicator is our 260, 280 nanometer ratio of these two different peaks that we have in each of these bottom two graphs. And we can see that the ratio is the exact same between our VLPs and our native PVX, which is 1.2. The last thing I'll point to is a size graph of the lengths of our VLPs as compared to VLPs made with gRNA, like we did previously. So just recombining the components of PVX in vitro, and then our native PVX. So you can see native PVX on the far right has a really tight distribution. It's almost like a solid line. Whereas as we move to VLPs, we get this expansion of polydispersity, even though we have the same components, genomic RNA and coproteins. And then you can see a size shift down when we assemble our eGFP SL1 VLPs. So we'd expect this because the mRNA is, again, a fraction of the size of the genomic RNA. But we do have some overlap because, like we said previously, there are some higher order assemblies. There's likely some genomic RNA contaminating the eGFP SL1 VLPs. So that's why you see those error bars. But we can say that there is some more evidence that it is RNA transcript dependent, the size of these particles. And then here's just some TEM of what those particles look like. So the top row from left to right is just 3 different magnifications of our eGFP VLPs, then our gRNA, and then our wild-type PVX. So again, similar morphologies. The eGFP SL1s tend to form more of a rod shape on average, but they do form some higher order assemblies that border on being filamentous, like the bottom 2 conditions. And so we also wanted to show some transfection data. And before I get into it, I just wanted to show you the gating strategy for the flow cytometry assays that we did. So it's pretty simple. We just did 3 main gates. We did all cells, basically all cells that were not debris. Then we gated out the doublet cells. So these are cells that are stuck together because we want to just have single cells in our data. And then finally, we did a bifurcate of our negative population of cells for GFP versus our positive. So what this means is that we have some cells in our population that are green and some that are not green. And then we did this as a time course, so you can see how this changes and how these populations change over time from 3 hours all the way up to 72 hours. And here you can see in blue, that's the negative unstained population. And then in red, you can see the GFP or the green population changing over time. And the profile looks very similar to what you'd see with native GFP or a lot of fluorescent reporter transfection studies. And then how did this look in terms of efficiencies? So we did a lot of different conditions from left to right in the upper hand plot, stacked bar graph. You could see that we took VLPs at 200 micrograms treated in a 6-well plate, which is a lot of VLPs, but they're very biocompatible and they're not cytotoxic, as I'll get into in a little bit. And then we did different conditions where we compared them against positive control mRNAs that were transfected with lipofectamine and VLPs that were treated with RNAs after assembly. So essentially, we had VLPs that were lipofected with lipofectamine. We had them first treated with RNase R and then lipofected with lipofectamine. And then we had the RNA controls for that. So RNA treated with RNase R, which has no expression, and then a couple different conditions of RNAs at 1, 2, and 4 micrograms of mRNA as comparisons to those different amounts of VLPs. So we can see that we get pretty good expression. And our best one, the non-RNase treated one, we have upwards of 80% of positive cells. But for the RNase treated one, which is in red, the third from the left, we see somewhat comparable, but we get drop-offs as we get lower in those microgram amounts of VLPs. And in the bottom right hand, you can kind of see those dosage conditions for these 6-well plates. So for every average of 10,000 to 50,000 cells, you can see a dosage increase and a dose escalation from 100 micrograms of VLPs to 300 VLPs without a drop in the average number of viable cells per well. So what this is basically showing is that we can dump a bunch of VLPs onto our cells. And despite the rates of encapsulation or lipofection, we can get really good outcomes in terms of biocompatibility and no cell killing or infection or contamination. All right, and then this is just a set of micrographs to show you what that looks like visually. So you can just see that over time, across these different conditions, from 0 to 48 hours in this case, we get increased amounts of GFP expression. Probably should have increased the contrast for a presentation on a projector, but on my screen it looks a lot better. And then we also wanted to just make sure that if we disassemble these VLPs, do we find those mRNAs in the extracted RNA from those disassemblies? So we took these VLPs, we ran them back through that disassembly process. We purified away the mRNAs, and then we ran RT-PCR. So what RT-PCR basically does is it amplifies RNAs based on basically a KIT and a fluorescent reporter molecule. And then the faster something amplifies, the more of that RNA is in that sample, right? So again, in the bottom left hand, we benchmarked against just regular EGFP mRNA, no SL1. We did our EGFP SL1 mRNA, and then we did 3 different conditions that we saw on the previous slide for VLP. So 300 micrograms, 200 micrograms, 100 micrograms. And if you go back to the beginning of the presentation, how much RNA is in these different conditions? Well, every 50 micrograms of VLPs, remember, has 1 microgram of mRNA as a comparison. All right. So to wrap up this part of the presentation, we've shown that we can get different, we can get synthetic mRNAs incorporated into our VLPs, and we can deliver them successfully into cells, given certain conditions. And they're able to be expressed in mammalian cells as well, which is a good step towards proof of concept. The PVX origin of assembly site is still working, even though we've moved on from gRNA and gRNA fragments. And we are getting similar morphology to our earlier studies with rod and filamentous shapes in our VLPs, and that these delivered mRNAs are capable of producing that EGFP protein, despite having a lot of other features from PVX, like the UTRs and the origin of assembly RNA stem loop, and that these VLPs can protect their cargo from things that are endogenous in the environment, like RNase R and other nucleases. All right, and the last objective was kind of a fun one that happened at the end. This was just the last few months of this PhD, but it was pretty exciting, and it was kind of a moonshot idea. We had already shown in things like TMV that we can get encapsulation of different RNA classes, like long non-coding RNAs, small interfering RNAs, microRNAs. These actually are encapsulated relatively well by VLPs across different virus classes. But one that nobody's done before is circular RNA. So could we make a ring-shaped VLP? And let me be clear, this does not exist in nature. There is no virus that we know of naturally occurring that makes this shape, which is kind of me spoiling it a little bit because it did work. But let's talk about what is circRNA. So circRNA is kind of this naturally occurring thing that happens in a lot of eukaryotes, bacteria, and archaea, in that when you have canonical mRNA production in cells, they produce things like I've been showing you. They make a pre-mRNA transcript from DNA, they cap it, they tail it, and that forms linear RNA, right, on the left hand here. But what can also happen, given certain features on an RNA, is something called backsplicing. So backsplicing is what happens when you have introns and extrons, a pair of each on either side of an RNA, and they essentially will be spliced by an enzyme like a twister ribozyme, and they can be religated back into a circular form of that RNA. So sometimes they exist solely as a circular version of these RNAs. Sometimes there is both a linear and a circular version, but they have a lot of different use cases, and we're just starting to learn about them the last few years. And so obviously they can make protein, but they can also serve as a way to interfere with other genes. They can be regulatory, and they can shut down different genes and different RNAs. And the great thing about them is because they're covalently linked into a circle, they cannot be degraded canonically by the nucleases that you typically see degrading RNA. One being RNase R. They are somewhat degraded by things like RNase L and different ones that cells and different bacteria produce in response to a viral threat. So they're actually treated by a whole different pathway for the most part as compared to linear RNA, which is really good because the problem with using mRNA therapeutics, as we've seen so far, is that we inject them and they're very short-lived, right? Which is really troublesome when we're trying to redose, when we're trying to control the efficacy of our therapeutic. And in this case, we're able to get a longer expression window and timeline, hopefully we can get better outcomes for patients. All right. So how do we design this? It's I have to give a shout out to the Molly Lab. Prashant was a very unlikely but very welcomed collaborator. And one of his PhD students, Michael Tong, I should give him a shout out at the end, helped me design this. But it was from their recent Nature paper where they essentially took a circular RNA and used a lot of different new techniques. I'm not going to get into the mechanisms of each, but they involved having this twister ribozyme flanking sequences, two ligation systems, and then those split group introns and extrons on either side of the payload, which basically means that the twister ribozyme will help take pre-circ RNA in this middle section right here and ligate away the free introns and the payload into those two products right there. So we'll have, as you can see on the tape station of the bioanalyzer, without treating with RNase R, we get three products. We get free introns, basically things that are the ligation system and the ends of those split group introns. And then we have our payload being backspliced and ligated covalently to form circRNA. But then we also have ones that didn't undergo that circularization, which are just these pre-circRNAs or like linear transcripts that are what are used as a template to make circRNAs. So when we've made the circRNAs, we basically treat all of that with RNase R because it gets rid of any linear RNAs, which is those introns and that pre-circ RNA, and we just get one band of circRNA at the end of this production. And one thing I will say is that our previous study, we used eGFP behind our SL1. We did several different iterations here, and we actually moved it in front. This is actually a little bit off, I think. And yeah, so the position of your stem loop structure actually affects gene expression, which is interesting. So that stem loop structure that's necessary for making VLPs interferes with polymerases, ribosomes coming in and making protein from your mRNA, which is something I haven't seen too much literature about. And here's kind of the money slide. So we were able to take circRNA. Here's a TEM image of circRNA. Again, not very many images of that for TEM, but I don't really know why someone would take a TEM of circRNA. And we see kind of this intermediate form, partial VLPs, where we see the beginnings of nucleation to form a circular VLP. And then we get this ring-shaped circ-PVX VLP, which was kind of like a cool moment in the TEM core when I first saw this. But you can see that it's kind of this same shape as the input circ RNA, and we get this like very high fidelity size control when we make circ VLPs, because again, we have different things happening with linear VLPs where we have multiple incorporations of RNAs and stuff like that. But in this case, we're kind of confining the system to just assemble around one molecule. So again, here's just kind of what the size looks like. The elution profile is very similar, but a little bit right shifted from PVX and the VLPs we saw earlier. There's actually a little bit of a double peak here, and there's some intermediate peaks down here. And I think those are just some intermediate VLPs, some partial assemblies, and some free particles or aggregates. And on the right side, you can see the size difference in terms of diameter, not length, but diameter between circRNA and our circ VLP. So we do get this huge shift and a pretty tight distribution compared to our other VLPs of our circ VLPs versus circRNA. And a lot of them cluster around about 200 nanometers in diameter, which is interesting because the circRNA, for the most part, as measured by TM, had around 300 nanometers in diameter, meaning that we might have even greater packaging efficiency when using circular RNA for making circ VLPs. Well, that's TBD. And then here's the transfection efficiencies and the confocal micrographs of the different conditions. So again, same strategy. The main difference here is that there's not a currently commercially available enzyme that degrades circ RNA. These studies are still so preliminary that when we do transfection studies and when we try to incorporate them into nanocarriers, those are very few and far in between. And in order to even attempt to degrade circ RNA in an in vitro environment, you need to basically get RNase L, polyl ornithine, and then activate it through these small oligonucleotides to get the RNase L to actually cleave this molecule. And only one lab that I know of in Maryland has done that. It's very complicated, and it seems like it doesn't work that well. But it's great for me because I get to say that I didn't need to treat it with RNase. I did try. I did get RNase L and try to activate it through some oligonucleotides, but I didn't have poly L, and I don't think it would have worked that well anyways. But all that suffice to say is that we still get non-inferior transfection efficiencies between naked lip-affected circRNA versus our lip-affected circ VLPs. All right. And just to kind of wrap up this story and these projects, we've shown that we can get different morphology. So here we've really shown shape engineering, which is a big tenet of immunoengineering. When we talk about metal nanoparticles, PLGA, different materials, we like to mess with the shape, the aspect ratios, because those give them different functionalities as materials. And here we're showing it with a soft matter virus-like particle, which is pretty cool. It's been done before, but we haven't yet seen anything that is ring-shaped. I like to say halo-shaped because I think the marketing is a little bit better there. And we've shown that we can also deliver these into mammalian cells. It actually worked a little bit easier and took fewer attempts, so it might be a better morphology for getting into cells. And if we had more time, it'd be great to do a longer transfection experiment where we go maybe 10, 12 days after transfection to see that green signal from EGFP being carried on. And yeah, one thing that we saw is that we overcome steric hindrance. So when I was talking to Michael Tong from the Molly lab, one concern that he brought up that I also had was that stem loop structure, that little loop that we've been using all these projects, will actually prevent the circRNA from forming and ligating into that backsplice form because it'll bump into that loop. It's a physical structure that's getting in the way. But it was just small enough to make these circRNAs. That was the first hurdle. The second was, okay, is PVX filamentous enough to bend into a circle? And here we showed that that's also possible too. So we not only get mixed assembly and overcome the steric hindrance from small proteins bound to the surface of coproteins, we also overcome steric hindrance in terms of things bumping into each other and forming that circular kind of halo shape. And they're a little bit more monodispersed. That claim is a little bit outblown, I would say, but they do exhibit a smaller diameter than circRNA, which is pretty interesting. And just to wrap things up, like Nicole mentioned, I made a couple things in lab. I made Biomark QA2, and I worked with the folks at Future House, a nonprofit in San Francisco, to make this. But it's basically a literature query tool. We tested it against subject matter experts for finding biomarkers in literature. So we had some postdocs and PhD students do it the old-fashioned way. I did it in about 10 minutes, and you can see all the different markers that you get from a question. So here we asked it about, does the intratumoral injection of CPMV, in that instance, what different cell types, what proteins, what genes are upregulated? And here you can see a breakdown of all those different biomarkers, their clinical significance, the literature, and the page from which that information came from, and it saves you a bunch of time. And then the last thing is virtual PI or postdoc. It's made in the likeness of Nicole, but she has to grow out her hair a little bit. And it's called Little Steinmetz, and basically it's a way of doing automated experimental design, or advising, I would say, not full design, and mentoring, because it can give you some advice. Yeah, yeah. And if you're asking what I'm doing after the PhD, professors that want one of these, it's $1,000. Just let me know if you want one. I'll make a nice logo for you in your likeness. Like Nicole said, a couple of papers, a patent, a few conferences, and yeah, thanks to everybody in the lab. And he's not on here, but thank you to Michael Tong. That last project would not be possible without Michael. And a special thanks to, of course, my PI, Nicole Steinmetz, and my mentor, Krista Barkovich, and my two students, Megna, who is here today, and Gotham, who is too busy getting like every industry position under the sun and doing co-ops every year. And with that, I'll take any questions.
For some questions from the audience.
Yeah.
Do you have, for the circular RNA and circular PDX, do you have like, is there another way to validate the circular integrity with a different experiment than the TM?
Yeah, that's a great question. So was the best with the time and resources we had for this project. We're also trying to send some out for cryo-EM. That gives us the atomic structure of the virus-like particle. We'll see if that works. It's a little bit of a juggling act in terms of stability, so we'll see if we can get a batch out. But besides that, typically you could do things like dynamic light scattering. PVX is unique in that it's a filamentous virus, so when you hit it with light from the same angle, it's bouncing off at different angles, and the photoreceptors in the DLS are going to give you different results every time. So anything with high aspect ratio is really hard to measure in DLS, whereas a lot of our lab can use it for things that have radial symmetry, like icosahedral viruses. Otherwise, I would have done that as well. But it didn't work for Circ PVX either, even though it's like a slightly different shape. It was just too wiggly and has a hole through the middle. Yeah, and that's the other thing too, and we've talked about this too, is that we could do immunogold staining on this, and just show through binding of metal gold nanoparticles the outside that we do indeed have coprotein in there, and that it's coating it at all the different angles of the ring. So that would be another way of doing, like, taking the TM even further and confirming that we have Circ VLPs. It's a good question. And then Zuha?
Very impressive. Thank you, Brian. Yeah, I think the immune goals might help the manualize the RNA, make it visible, and especially for this one. Yeah, so my question is, what cellular internalization pathway do you believe are responsible for the output of the circular RNA?
It's a good question. So because we used liposuction, it's kind of the canonical lipid-mediated internalization and degradation pathways for that. We can do different studies for that, and we've talked about it. We just didn't get around to doing it for a couple of these last studies. But there are ways of using different dyes and kits and antibodies to show which pathways internalize the VLPs by themselves versus in combination with lipofectomy. The other thing I didn't show today too is that we do get small amounts of transfection without the assistance of lipofectomy. It's just so low and needs so much optimization that we thought it would be best served for a different paper down the road. But like I said in the beginning, it does have an affinity for binding to B cells, and so there could be some kind of internalization mechanism there. It could be glycoprotein mediated, but that's still something we have yet to find out, and would be a really cool mechanistic study. Yeah.
Oh, you brought this up with the circular RNA and stuff that it's like, well, probably more stable than your typical mRNA for therapeutic. Is it because of, I might have missed or just misunderstood, are there different RNases in mammalians, like those, are there different levels of those RNases in the mammalian cells that would make that circular RNA last longer, or they're not present and so that RNA would stay there longer?
Yeah, it's really interesting. A lot of these nucleases are really evolved to recognize linear RNA. And if you go back far enough, the earliest viruses are actually just a piece of circular RNA. Before we had coproteins and capsids, if you look at like viroids, which are a circular RNA virus that infects plants, it has no proteins. It's just naked RNA that's just floating around producing more of itself, right? And we just actually, this past year, found that in the human gut. They're called obelisks, and they also kind of float around. They're not infectious, though. They just infect the bacteria in your gut. But to answer your question, even hex cells and BHK21 cells that I showed in this presentation, they create activated RNase L or activated RNase R in response to things that they perceive as viral. So there's actually a lot of it floating in the dish all the time when you do these virus studies. When you talk about an in vivo system, which is hopefully where we're moving towards next with a lot of these studies, you just multiply that even like morpho, like blood has a lot of serum nucleases per like liter, per milliliter, and they're kind of everywhere in the body. They're everywhere in the environment. But the reason that circRNA is so stable is that very few of those recognize circRNA because it's rare. The abundance of it is really small and compared to like linear RNAs. And a lot of times it's good that it's not degraded because it serves as a regulatory molecule. But basically you can think of it as like a little factory that's just producing little linear transcripts that are just making protein. And so those will make protein while the parent molecule is just kind of like okay for I think up to 17 days, 18 days. Whereas like traditional mRNA is like, if you're lucky, 4 to 7 days. Yeah, sure, yeah.
And then with the cert PBX, would you expect that to work as well as regular PBX? I mean, like we showed efficacy in, animal models with PBX. For reducing tumors.
Oh yeah, yeah. I mean, I can't speak on how well it would work, because I can't. It might utilize the same like cytokine activation profiles and pathways as PVX, but it might have different tumor and tissue homing, drainage, like lymphatically, and some weird physical characteristics like that, just because the shape is different, right? That's an interesting question. I will say though, in my example here, they're pretty small, like circles. So maybe it's a size dependent thing where like small circles penetrate better versus bigger. I would assume it'd be something like that.
Our next student to solve.
Yeah, next person, Magna.
Let's thank Bryan again. And then we will use the public. Recommend you will stand.
Stop recording.